Welcome to ASM Live at the 115th meeting of the American Society for Microbiology. We're happy to have you join us here. We're in New Orleans, Louisiana. I'm Stanley Malloy. I'm your host today. And joining me today is Lauren Aber. I invite the people in the audience, the live audience, to come up and ask questions at the microphone. And any of you listening from your computers or online, please send questions at hashtag ASM Live through Twitter, or you can send questions from the ASM Facebook site. Today, we're going to talk about some questions that are important, interesting, and sometimes just downright surprising about new things we're discovering in microbiology. And Lauren's going to tell us today about some studies that she's been doing that relate to our life in a fecal cloud, a fecal aerosol. It brings such nice connotations, doesn't it? Could you tell us what your research involves?
Thank you so much for having me. And I'm working at Quinnipiac University currently, and we're looking to study the transmission through a fecal oral route of potentially pathogenic bacteria that are aerosols created from the toilet and how they might end up on your toothbrush.
Aerosols created from the toilet. Is this something we should worry about?
Definitely. I mean, it's not something you would typically think about, but especially in a communal bathroom when you don't have a lid most likely on toilets. And studies have shown that aerosols can spread about 6 to 9 feet when the toilet's flushed, and all those little droplets of water that come out of the toilet can contain bacteria and viruses.
This is a nice image to have. So in your study, you showed that you could get contamination of toothbrushes?
Mm-hmm. So we've been collecting toothbrushes from students at Quinnipiac, and we're just asking them at the time of the collection to donate their toothbrush. We haven't specifically given them a toothbrush or anything. And then we also collect a survey asking about their hygiene habits with their toothbrush, how they store it, where they store it, and also if they sanitize it, whether that be cold water, hot water, using mouthwash, and then also if they have any illness at time of infection, oral illness or gastrointestinal illness, to see if that's going to impact what we find.
Do you give them a new toothbrush? Or
Yes, we do. Yes.
Those poor students.
Yeah. No, we do offer them a new toothbrush at the time of collection as well.
And what have you found?
So from what we found, we're growing up first we decapitate the toothbrush heads, put it in a phosphate buffer solution, and then grow it out on blood agar, both anaerobic and aerobic, McConkey agar, which is going to be selecting for our gram-negative bacteria, which is really what we're looking for. And then also Mythbusters agar, which is going to be showing what's going on currently in the mouth, just looking at the normal oral flora, and then also TSA to get a general growth medium. And we're seeing lots of different bacteria growing on toothbrushes, but we are finding a significant presence of fecal coliforms on toothbrushes. About 54% of toothbrushes that we've collected had some growth on McConkey agar.
That's impressive. But those poor toothbrushes you decapitated.
It's a strong word to use, but we do take the head off the toothbrush just kind of with a pair of scissors, and then they snap pretty easily, and then put them in the solution and vortex to elute off the bacteria, but without harming them. From other papers that we've read, that seems to be the most effective but not evasive method to remove bacteria from toothbrush heads.
And so what kind of difference do you see in the communal bathrooms that you've looked at versus what you would expect in a bathroom that's used by fewer individuals?
So when we've looked at these toothbrushes, all of our bathrooms on an average were used by 9 people, and that varies based on the population of students that we're taking the toothbrushes from, that some students were sharing, with 2 or 3 people, usually as they increased in grade level at the school, because they weren't living with as many people, but we're also collecting from dorms where there's one bathroom on the floor and there's lots of people. We didn't look at specifically a difference between what each bathroom, like how many numbers of people to what was growing on them, just an overall total that there was a lot of growth. But we did look at the difference between how people stored their toothbrush and what grew on them. And even if you use a closed cabinet, which is recommended by the ADA, that in a communal bathroom, you're still going to see fecal coliforms on those toothbrushes.
So they can sneak in past the edge of the cabinet. Huh?
Or you're potentially self-contaminating when you touch the faucet, which remains in the sink at all times and doesn't get sanitized every time you turn on the faucet or not. And that can end up, and they can also, especially in communal bathrooms, when you've got the door handle coming in and students that take their toothbrush outside the bathroom and store it outside, they're touching all the surfaces of the bathroom as they come into the bathroom. And when you're not leaving your toothbrush in the bathroom as well, you're placing it somewhere potentially on your way in to brush your teeth.
So there's not an easy way to get past this fecal cloud.
Right, yeah. So I mean, the best practice is to store it in a closed cabinet and also to not use a toothbrush cover. A lot of people do think that a toothbrush cover is the best way to prevent anything from getting on it, but you're really keeping it in a moist, dark environment, which is going to promote bacterial growth on the toothbrush, actually.
Well, we have a question from the audience.
Yes, this question is from Janet Rowe of ASM. She asks, have you thought about looking at comparing your participants' dental health histories in your results?
We haven't looked at that. That would be interesting to look into.
But one of the things you said was you looked at their general health, their self-reported health.
Yeah.
Do you see a correlation with that?
We really only had a very few percentage of people, I think it was 3% that said they had some type of illness going on, and they didn't specify if it was viral, bacterial, or even if it actually had been diagnosed, or if they just thought, you know, they were coming down with something. So it's hard to really tell what. And we only had a couple students report that there was gastrointestinal illness, and we looked at what those specific bacteria turned out to be, and they were commonly known foodborne illness. They're bacteria that cause.
So in addition to the coliforms, what other kinds of microbes do you find on a toothbrush?
So we're finding a lot of bacteria that you commonly find on the skin or found in tap water, like a lot of Pseudomonas, and then also your oral flora is also on the toothbrush.
So the oral flora can persist on the toothbrush for a reasonable period of time?
Yes. And we're also collecting the toothbrushes and sampling them within 12 hours of last usage so that to prevent them from drying out before we sample them.
Just from curiosity, one of the things you said was that some of the students used mouthwash to try to decontaminate their toothbrushes. Any impact of that?
Actually, no. From what I found, there was 2 students that from our smaller sample size, I mean, it was a very small sample size, that regularly said they use mouthwash to decontaminate their toothbrush, and both of those had fecal coliforms on it. So, I mean, that we didn't ask what type of mouthwash or anything like that, but it didn't seem to have an impact on the presence of eliminating fecal coliforms from the ability to grow, which is kind of interesting and alarming.
I'll bet if this news became widespread, there would be a great market for disposable toothbrushes.
Yeah. One study I actually read recommended that you change your toothbrush as frequently as possible, and they said, we understand a daily toothbrush is not feasible. But
I understand that the Mythbusters took a look at this idea at one time. Could you explain what they did?
yeah, so they placed toothbrushes around a bathroom and looked at to see where aerosols really end up, and they really do travel across the bathroom, that you are seeing the spray and the aerosols get on all surfaces in the bathroom.
So from distances beyond the bathroom as well?
I'm not sure about that. So from what I know, usually about 6 to 9 feet is how far they go, and communal bathrooms can be larger than that. So in our study, we do ask, measuring on the idea of a body length is 6 feet. Do you store your toothbrush an arm's length away, less than a body's length, or greater, to see? And all of those seem to have contamination.
We have another question from the audience.
Yeah.
Yeah, I have 3 or 4 questions here. So Steve asks, Steve is on Microbe World, and he asks, have you looked at any sanitization and whether these colonies will grow on McConkey agar afterwards?
Okay, yeah. We ask the students if they sanitize their toothbrush when they regularly use it, and the sanitization that we ask about is rinsing with hot water, rinsing with cold water, if they use a UV sanitization method, or if mouthwash is regularly used. And we didn't find anyone that used a UV sanitization as a college student, so we couldn't speak to how that works. But it seemed that hot water and cold water didn't really have a different effect of the total bacterial growth on the toothbrushes. But as I mentioned, that mouthwash does, from the couple samples that we had that people usually used mouthwash to sanitize their toothbrush, fecal coliforms were seen, and we were measuring that by presence of growth on McConkey agar, because that would select for an enteric bacteria to grow.
Great. Another question was from Misa, also on Microbe World. She asked whether this result has been published?
Not yet. So I think it's something we're looking into.
Great. And then another question we had was, do any toothpastes have any antimicrobial substances that might help with this issue at all?
I think there's been some research on it. We didn't look at the type of toothpaste that students were using, but that's definitely something to consider, that it could have an effect, and definitely, especially with the content of it.
Great, thank you.
Presumably you could take your toothbrush and dip it in Clorox, right?
Yeah, I don't know how that's going to harm your gums when you...
Well, no, then you rinse it off afterwards, hopefully.
Yeah. I did read one study that looked at vinegar as a non-invasive method of sanitization, and that seemed to be reasonable, but you definitely want to get that taste off of before you. So I think you've got to have a balance between what's going to happen to you after you sanitize it, and to really make sure it's not allowing for any growth to occur, but also not harming you in the process.
You know, this is aside from what you've done, but it's an interesting thing when you look at all the people who've sampled different parts of a bathroom for fecal coliforms, and come very often to the conclusion that people spread them by their fingers. Whereas, in fact, I think from your studies, it may be that some of them are associated with an aerosol, not directly spread by the fingers. Of course, then the fingers can take them even further.
Yeah, and I mean, definitely, and even if they're spread by aerosols, you can then transfer them to different parts of the bathroom, especially, or if aerosols are landing on things and then, again, you're picking up your toothbrush, you could infect yourself, or flossing, you know, your hands are getting in your mouth, and that could be another self-inoculation.
Lauren, has this changed your practices in terms of toothbrush?
Well, actually, I sampled my toothbrush a couple times throughout the study, and was happy to find that I didn't see any fecal coliforms, but it's definitely made me very aware of how I store my toothbrush, and also thinking back to, especially when you're living with a family growing up, you have a shared toothbrush holder, and you really don't think of your family members as having bacteria that you don't want to interact with, but a very easy way to transfer them. Most likely in college settings, people aren't sharing a toothbrush holder with their roommates, but especially in family settings, that can be an easy way to transfer something, you know, like strep or something like that, or any type of enteric bacteria is a little droplet of water that you spray as you're trying to put your toothbrush back in the holder, you knock them, and that's because you can definitely see cross-contamination there.
I mean, even the normal flora from your mouth, like the different types of Streptococcus mutans that have different impacts on dental and systemic health.
Yeah, I'm not sure exactly how well dental, like, they would survive in your oral, like, based on your own oral flora, that they might get rid of the new bacteria that you're introducing, but especially with enteric bacteria that are not commonly found in the mouth. And something interesting that I read, about 80% of the fecal coliforms that are present on a toothbrush actually aren't from you when you're living in a communal bathroom. And so that's kind of alarming that those are the ones that are potentially going to make you sick when you don't have them already in your gut, that you would be able to have a response to them.
If you're talking about an average of 9 people in the bathroom, and the bathroom being an aerosol thing, about 80% is about what you would expect, just by chance.
Yeah, that's
right?
very true. And especially in a setting like that, when you've got a moist environment in a communal bathroom, that in a college, everyone's got different schedules, that people are showering all day long, keeping the bathroom very humid and allowing for a good environment to promote bacterial growth.
So what would you do next if you were going to carry this study on?
I think it'd be really interesting to actually give students toothbrushes at the time of, and like really monitor exactly how long everyone's using the toothbrush and then continue with the same, but also add a few more questions to our survey. And I think also at the time of collection, swabbing someone's hands to see what they're potentially introducing so that we can more definitely say that the bacteria on the toothbrush are coming from an aerosol or another source, that you're not inoculating them with your hands as you're brushing your teeth.
I'd like to just remind the audience, please join us by asking questions at the microphone if you're here live. And if you're listening online, you can send any questions via Twitter at #ASM Live, or you can ask the questions at microbeworld.org or at the ASM Facebook site. We now have another question from the audience.
Yes, this is again from Rich on Microbe World. He asks, besides the fecal coliforms, do you have a method of determining the burden? And then he also wanted to know if there were, I think you mentioned this, but there were fewer coliforms on toothbrushes that were stored in the cabinet versus ones that were stored in the open?
So we're looking at at the time of collection, we ask a survey, and it's self-reported, of where you store your toothbrush, whether it be on an open shelf, a countertop, or in the cabinet. And then we also compare that to the number of bacteria that are present on the toothbrushes. And we are seeing that it is recommended the further away or when it's closed, you're going to see less growth on, especially if the toothbrushes have the time to dry in between usage. But as far as the burden of bacteria goes, we haven't, you can see bacteria present on all toothbrushes, and studies have shown that after one use, you're going to see the presence of some bacteria on them. So.
Not all bacteria are so bad.
No, and a lot of them are, you want to have normal oral flora, and you don't want things that are causing caries and such, but there's going to be normal flora that are taking care of the potentially pathogenic bacteria that you might be introducing, that it is healthy to have some, and that's completely fine, but we want to see what those are and then what we might be introducing.
If I understand this right, when you brush your teeth, a lot of what you're doing is eliminating the biofilms that allow oral microbes to accumulate. You're not really getting rid of microbes per se, right?
No, and so when plaque is forming, it's really, as you said, like a biofilm that's adhering to your teeth. So you want to get rid of that, especially because if you leave it, it can build up to tartar and cause other sorts of dental diseases. But and also when you think about brushing, that people recommend a soft toothbrush, and sometimes dentists have to tell people that you're brushing too aggressively, but you can cause trauma while brushing, especially to your gum tissue. And if you've got something pathogenic on your toothbrush, you're introducing that directly into your bloodstream.
I think we have another question from the audience.
Hello, I'm Lisa Foster from Trinity College. And Lauren, this is an interesting study, and I was wondering, do you have any sense of how long these organisms that are contaminating the toothbrush from the aerosol might persist on that toothbrush?
That's really interesting, because we're looking at the toothbrushes right after usage, so we haven't looked at how long they're actually allowed to grow. That, from what I understand from our study, is that they've been recently inoculated, because in theory, with a dry toothbrush, especially without any nutrients present or water, the bacteria shouldn't be able to persist. So these we're introducing in a recent manner, and then you're directly brushing, and those are being introduced to your oral cavity.
So might a simple thing someone could do, would it be to just make sure that their toothbrush is dried after their use before they reuse it?
The best way to store it is upright in a facility that's going to allow it to dry fully in between uses. And when you're brushing your teeth twice a day, the distance between the two brushings would allow the bristles to dry in between usage.
You know, a follow-up to this question is very interesting because I think your answer is based on the oral microbes that survive on the toothbrush. And in fact, some organisms like certain fecal coliforms or other things that could be aerosolized might survive a little better on the dry, inanimate surface.
Yeah.
And so it would be interesting to know that. I mean, it gets to be a very complicated problem. You know, one of the things is this question of when the aerosol happens, what's the relative abundance of microbes in that aerosol, right? Because the toothbrush is such a small little surface out of the whole bathroom, and for you to get detectable numbers of fecal coliforms on that toothbrush suggests that the air in that bathroom would have a very large number, right?
Definitely.
So if you just took a McConkey plate, you open it up in the bathroom for different periods of time, you could potentially get a feeling for what those numbers are.
That would definitely be interesting to add on to the study, and especially if we're collecting all the, you know, a certain number of toothbrushes from the bathroom or the bathroom that we're collecting the toothbrushes from, to see and compare a colony count of what you're seeing on the toothbrush in comparison to what's growing on the McConkey agar. And see if we're getting the full spectrum of each toothbrush, or if certain toothbrushes in certain locations are really only getting one type of enteric bacteria.
You know, the other potential solution is just not to share bathrooms with people.
Everyone have their own bathroom in college.
Yeah, it might be a little more expensive, but. So you are going to go on from here and go to dental school. Could you tell us, what is it how did you get interested in this process relative to thinking about dentistry as a career, and where you think you're going to go?
So I got interested in dentistry when I was about in 6th grade, and my knowledge of dentistry has evolved since then. And I'm really interested in the connection between your overall health and how that impacts your oral health and vice versa. So I think that kind of influenced how getting involved in this research lab and looking at the microflora on toothbrushes. And then from that, really seeing, because more I've learned about different bacteria, especially normal oral flora, what impact that can eventually have on, and things like strep as well, that can have other, if left untreated, can have other effects on your body.
I think it's really interesting how our knowledge of microbiology, since the advent of our ability to look at population structures, it's totally changed so many fields, and dentistry is actually one of those fields.
Yeah.
I mean, the idea that all dentists for a long, long time have learned about oral flora, but many of the questions that you can now think about relative to microbiomes, and the kinds of questions that are natural extensions of what you've done, I think really wouldn't have been obvious questions 10 years ago.
Yeah. And now there's different studies being done on the exact type of toothbrush and style and the position of the bristles and how that's affecting a biofilm formation on a toothbrush head and potentially affecting the bacteria content and things like that, which is really interesting and new things that you wouldn't typically think about when you pick out a toothbrush.
It just shows how microbiology has infiltrated everything in a good way. So after dental school, what do you think you're going to go on and do?
I'm debating right now between going into general practice or oromaxillofacial surgery, but I definitely want to have my own private practice, and I think also working in a community health center for a little bit would be something I'm interested in.
We have another question from the audience.
Yes, both Rich and Misha on Microbe World would like to know, other than selection differentiation via agar, have you used any methods to identify specific microbes that grow?
Yes, we're doing biochemical testing. We do a TSI test, triple sugar, iron, agar, an oxidase test, and a nitrate test, which is all going to help us identify enteric versus non-enteric bacteria. And then after that, we're doing 16S RNA sequencing and running those sequences back on NCBI BLAST to get the exact strain of the bacteria we're looking at.
So you have these samples stored, and you'll be able to go back and re-assay the samples that you've already looked
Yeah,
at.
so at the time of collection, we're growing them out on the plates, and then we're gram staining as well, but we'll store them in the freezer, and then later throughout the semester in big groups, we would do the biochemical testing together, and then later on went back and regrew them and did the DNA extraction.
Another question from the audience.
Yes, Jordan on Facebook wants to know if you've done any studies to investigate the transfer of infective viruses.
No, we haven't looked at viruses, but that's definitely something that they're present on your toothbrush. And if you think about things you hear, like norovirus spreading, the very few number of particles that you need to transfer norovirus, that's an easy way if someone comes back to your dorm sick with that, and the whole way your whole floor can get sick.
I think norovirus is maybe an exception because it's so contagious that all it takes is one doorknob to spread it around. But there's a lot of other viruses that can also cause a variety of diseases and will stay in the aerosol longer and potentially survive better as the toothbrush dries out, which is an interesting perspective.
Yeah, I read one study that looked at, as you were talking before, about what's coming out of the toilet and looked at bacteria and viruses and how they elute out of the toilet. And that was pretty interesting to think about, that the porcelain might actually absorb the bacteria and viruses, and then after continuous flushes, you are still eluting out bacteria, even though when you look in the toilet bowl, the water looks clean.
See, this is another argument for outhouses.
No flushing to create aerosols.
No flushing required, right? Well, it's been really nice talking with you. Very interesting topic, and something that can change the way we can think about communal bathrooms for the rest of our lives. I want to thank the audience for joining us today. Those of you who are here in person and all of the rest of you who are watching online, please come join us again for the next episode of ASM Live from the 115th General Meeting of the American Society for Microbiology here in New Orleans. Thank you very much.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.